4.5 billion years ago, the Earth's atmosphere was entirely devoid of free molecular oxygen. The biological process that ultimately saturated our skies and oceans with this gas is the primary redox reaction driving the planetary biosphere. Photosynthesis is the mechanism by which photoautotrophic organisms, including terrestrial plants, eukaryotic algae, and cyanobacteria, harvest the electromagnetic energy of sunlight and transduce it into stable chemical bonds. This diagram outlines the macrolevel stoichiometry of the process. 6 molecules of carbon dioxide and 6 molecules of water are converted, via the input of solar energy, into 1 molecule of glucose and 6 molecules of molecular oxygen. While this equation is stoichiometrically precise, it summarizes a sequence of dozens of enzyme-catalyzed steps distributed across specialized cellular compartments. These pathways evolved over billions of years to manage the high-energy intermediates created during the reaction. By utilizing solar energy to drive the synthesis of complex organic carbohydrates from low-energy inorganic precursors, photosynthesis provides the metabolic energy required by nearly all heterotrophic biological systems on Earth. 3.4 billion years ago, the first photosynthetic bacteria operated under strictly anaerobic conditions. Because these organisms used hydrogen sulfide as a primary electron donor rather than water, they generated elemental sulfur as a metabolic byproduct instead of oxygen. 2.7 billion years ago, cyanobacteria developed the biochemical mechanism to utilize the stable water molecule as an electron donor. This allowed phototrophs to inhabit any environment with liquid water, removing the reliance on localized sulfur or iron deposits. This timeline tracks the resulting accumulation of molecular oxygen in the oceans and the atmosphere. Geological evidence in ancient sedimentary rocks shows this oxygenation peaked between 2.4 and 2.3 billion years ago during the Great Oxidation Event. The evolution of photosynthesis continued through endosymbiosis, with eukaryotic algae emerging 1.2 billion years ago. By 475 million years ago, the first plants emerged in terrestrial environments, followed 50 million years later by the evolution of structurally sophisticated vascular plants. The modern leaf is the result of continuous selective pressures to optimize solar energy capture as atmospheric conditions and light availability shifted over geological time. To trace the reduction of carbon, we must move inside the mesophyll cells of the leaf and examine the internal architecture of the chloroplast. The chloroplast is bounded by an outer and inner membrane, which physically isolate the internal biochemical reactions from the rest of the cellular environment. Inside the inner membrane sits the thylakoid system. This network of flattened vesicles is organized into cylindrical stacks called grana, interconnected by stromal lamellae. This segregation physically isolates the tiny interior thylakoid lumen from the surrounding protein-rich stroma, where light-independent reactions occur. The restricted volume of the lumen allows for the rapid accumulation of protons. This segregation creates the electrochemical gradient used to synthesize the molecular carriers that power the cell. Within the thylakoid membrane, chlorophyll molecules utilize porphin rings to capture photons. Chlorophyll A and B are primary antenna pigments, focusing solar energy into the reaction centers. This graph shows these pigments absorb primarily in the blue-violet SORAT band and the red QY band, reflecting green wavelengths. Carotenoids act as accessory pigments to broaden usable light, absorbing 400 to 500 nanometer blue-green photons and transferring the energy to central chlorophylls. High solar irradiance can deliver energy that exceeds the processing capacity of the electron transport chain. If this excess energy is not managed, it leads to the formation of reactive oxygen species that degrade the photosynthetic machinery. Carotenoids protect the cell through non-photochemical quenching. In this process, the pigments absorb excess excitation energy and dissipate it as heat, preventing cellular damage. The light reactions begin when a photon strikes photosystem 2, triggering the photolytic oxidation of water into oxygen and protons. The energized electron transfers to plastoquinol, which migrates to cytochrome B6F, using the energy to pump protons from the stroma into the lumen. This active pumping creates a pH gradient across the membrane. As protons accumulate, the lumen becomes acidic, while the stroma remains alkaline. The pressure forces protons out through ATP synthase. Flowing through, they force the enzyme's catalytic head to rotate like a turbine. This mechanical rotation, photophosphorylation, provides energy to fuse an inorganic phosphate onto ADP, creating high-energy ATP. Finally, photosystem 1 absorbs a second photon to re-energize the electron, which is then used to reduce NADP+ into the carrier NADPH. Through this membrane-level choreography, the transient energy of solar photons is captured and stored in the stable chemical bonds of ATP and NADPH. These carriers move into the chloroplast stroma to power the light-independent reactions of the Calvin-Benson cycle. The cycle is driven by the enzyme Rubisco. It often accounts for 30 to 50% of the total soluble protein in a leaf, a concentration required to compensate for its slow catalytic rate of 1 to 10 reactions per second. During carbon fixation, Rubisco fuses CO2 with the 5-carbon acceptor RUB, forming an unstable intermediate that cleaves into 2 molecules of 3-PGA. Next, in the reduction stage, ATP and NADPH provide the phosphate groups and electrons to reduce 3-PGA into the sugar phosphate G3P. The oxidized ADP and NADP+ are recycled back to the thylakoid membrane to be recharged. In the regeneration stage, 5 out of every 6 G3P molecules produced are utilized to precisely rebuild the initial RUB acceptor, keeping the cycle continuous. This leaves a net surplus of G3P, the actual carbohydrate output of the cycle, available for the plant's growth and metabolism. The Calvin cycle is the primary mechanism by which biological systems convert inorganic atmospheric gas into solid organic structures. The surplus G3P is chemically unstable and is immediately partitioned into 2 distinct biosynthetic pathways. In the stroma, G3P is converted into glucose 6-phosphate and polymerized into insoluble starch granules. Storing carbon as starch allows the chloroplast to act as an osmotic buffer. It stockpiles energy without altering the stroma's solute concentration, which would otherwise inhibit the enzymes. Alternatively, G3P is exported to the cytosol and fused with UDP glucose to form sucrose, the primary carbohydrate for long-distance transport. This dual economy provides local energy storage via starch and systemic distribution via sucrose. Despite its efficiency, the system is limited by the oxygenase activity of Rubisco. In high heat or high light conditions, Rubisco can mistakenly bind oxygen instead of carbon dioxide. This triggers photorespiration, a process consuming energy and resulting in a net loss of fixed carbon. C4 plants evolved a spatial solution. They capture carbon in outer mesophyll cells and pump it into specialized bundle sheath cells. This concentrates CO2 around Rubisco and minimizes its contact with oxygen. CAM plants utilize a temporal solution. They open their stomata at night to store carbon as malate, then close them during the day to process that carbon, while preventing water loss in arid environments. These C4 and CAM pathways demonstrate how the photosynthetic apparatus can be reorganized at the genomic and structural levels to overcome specific metabolic constraints. Once synthesized, sucrose is loaded into the phloem and translocated to the roots, stems, and fruits, nourishing the non-photosynthetic tissues of the plant. The efficiency of this carbon partitioning determines the total biomass and carrying capacity of the global ecosystem. Modern bioengineering and agritech efforts focus on reverse engineering these metabolic pathways to create crops that are more resilient to shifting climates. Understanding the mechanics of this biochemical engine is central to managing our planet's future productivity. For more deep dives into biological systems, subscribe and click the bell for updates.